Agora a gente está ao vivo, Ângela.
Tá certo, ótimo.
Então, boa tarde a todos, gente. A gente vai dar início aqui ao nosso seminário com a Mariana, que aceitou rapidinho, né, Ângela, de pronto para apresentar. Muito obrigada, Mariana.
Obrigada, eu.
E
Mariana De Niz.
Mariana De Niz. Então, faça as honras, Ângela.
Não, você que é a host, você é a host. Eu sou só papagaio de pirata aqui.
Então, tá bom. Então, a Mariana De Niz, ela concluiu o mestrado dela em Londres, na London School of Hygiene and Tropical Medicine. Ela fez um doutorado na Suíça e depois um pós-doutorado em Boston, na Harvard School in Public Health, e em Lisboa. E atualmente é que eu não peguei o link, Mariana.
Ela ainda está em Lisboa, né?
Agora estou em Paris, realmente estou em Paris fazendo um visiting fellowship por um tempo para aprender a fazer Electron Microscopy, que é parte do que eu vou apresentar agora.
Tá,
muito
ótimo,
legal.
Bacana.
Então, agora, Mariana, eu vou colocar aqui a lista de presença para o pessoal ir preenchendo. Se vai entrar certinho. Seria bom a gente ter um feedback aí da plateia, se está tudo certo, vocês estão com som, estão conseguindo ver. Tem um boa tarde da Tainara aí, acho que está tudo certinho. Então, eu vou habilitar aqui a sua tela do seminário.
Ótimo.
E deixa eu ver, aqui é um momento tenso que eu vou só me fechar minha tela aqui e você já pode entrar no modo apresentação, tá, Mariana?
Ok, ótimo.
Então, deixa eu fechar aqui.
Ali já está bem, acho que é. Ok. Bom, well, I think I'll present this in English, then it is here, I think, for me at least to talk about science in English. So this is work that I did in in the second postdoc and the visiting postdoc that I've been doing at Institut Pasteur. I don't know how you see this particular panel here. I see a note of music, but it's supposed to be an animation, so I don't know why. But so I'll talk to you a bit about my work in these postdocs. I basically, since early in my PhD, my interest was in tissue tropism and a lot of all the biophysical aspects of tissue tropism of parasites. So early in my career, I focused on Plasmodium, and later I moved to Trypanosomes. And so, yes, this is the work. First, I will show you what I did in Lisbon all the time I was there, and later what I have been doing at Institut Pasteur. So I imagine you are familiar with Trypanosoma brucei. So it's a parasite that causes human African trypanosomiasis, and it has two defined stages of its life cycle. So one, it's in the fly, where several different forms have been very well characterized. Here we just show two in this particular panel. But yeah, basically it goes from the stumpy forms that are transmitted from the mammalian host to the tsetse fly, and then that turns eventually through many different compartments in the fly. They turn into metacyclic form that finds its way back in a different bite. And within the mammalian tissues, the parasites, usually the two forms that are well defined are bloodstream forms. Among the bloodstream forms, the slender forms and the stumpy forms. This panel that I'm showing is just to summarize what Luisa Figueiredo's lab found, that basically parasites can go to the adipose tissue preferentially. And so when I read her work and I knew about her, I applied to her lab with an EMBO fellowship, in fact, to study how parasites. So first of all, whether only the adipose tissue is preferred for tropism, and then how parasites can cross the vasculature. So all my career before that, I had focused on the vasculature of the host as a way of parasites to move within the entire host. So there are many things that I wanted to investigate. One was, so I divided it here and I will present it during the talk in these factors. So the first one is host factors that enable this traversal from the vasculature that you can see here to the tissue, and then separately what are tissue properties that are related to whatever happens in these tissues, and the other is parasite factors. So considering just the vascular factors, we divided it into different parameters as well. So the first one is whether there are specific, there's something specific about the adipose tissue vasculature that makes crossing in this place more favorable or better or easier than in other tissues. The second is whether it's not just the anatomy of the vessels or the density or the permeability, but rather specific receptors in those vessels. And the third is blood composition and rheology. So basically blood composition, by that what I mean is more leukocytes, as would be in the case of inflammation, more viscous blood, and the speed of blood flow among different types of vessels, so capillaries as opposed to arteries or veins. So the first thing I started asking, I immediately went to the adipose tissue, but I thought, this is interesting. Is it that only the adipose tissue gets enriched with parasites in the extravasculature, or are there more tissues? So we started doing bioluminescence imaging and intravital microscopy. Here I'm showing the intravital microscopy. So when we look into the vasculature only, we saw that some vasculature of some organs, so here in these heat maps, what we show is on the horizontal, the days post-infection, and on the vertical, the organs that we investigated. So basically we see that in the lungs, towards the end of infection, there's some enrichment, or the most enrichment, but in most of the organs, there are waves which are coincident with what we know happens with Trypanosoma brucei, that it comes in waves. We capped all these infections at day 20 because we do have some death of mice at day 20. So all this work we did in C57 mice. But when we go to see the extravascular parasites, so this is everything that crossed out of the vasculature, we see that indeed the adipose tissue was very highly enriched at various points, but the pancreas as well was very enriched. So, and we saw other types of parameters. So for example, the spleen is enriched early in infection, but not later. The same is true for the liver and so on. So we thought, okay, it's not just the adipose tissue. There are other organs we should be looking into. And I don't know if you're familiar with intravital microscopy. So this is basically a surgical procedure where you expose the organ of a mouse, and you can use different fluorescent markers. Either the mouse is a reporter line, which has some cells or tissues that are fluorescent, usually by GFP, or you can inject conjugated antibodies to an Alexa flu or fluorophore, or dyes that you can intravenously administer. The second thing that we showed was, I don't know why the actual image is looking a bit weird for me, but in my original presentation it looks different. So sorry about that. So the second finding is that there are many adipose tissues. So it's not just the gonadal adipose tissue, which is what Luisa had focused on. So we investigated many different tissues, and we saw that there are basically differences. So the summary of this slide is that the brown adipose tissue is less colonized than the white adipose tissue. And this is important because they have different metabolic roles. So different reservoirs provide different environments that are more or less favorable for parasite development. The third finding is what I showed you briefly in the first panels, where so here we classified by patterns what happens in terms of parasite distribution intra and extravascularly. So here you can see in blue the extravascular enrichment, and in red the intravascular. So we have a first group of organs where enrichment in the extravascular location happens from day 8 onwards, so that we defined as later in infection, but not initially. But it's nothing compared to the intravascular enrichment. So in these groups are the brain, the lungs, the heart, and the kidneys. And this extravascularization most often correlates with damage to the vasculature. So this is something I'll talk about a little bit later, but that's why it's only later. So early on, you find barely anything. The second group is also very interesting. So here you can see that these are lymphoid organs in their majority. I didn't include here the bone marrow. This we investigated later, and it has the same pattern. So basically there's enrichment early in infection in the extravascular location, but later on this disappears. And this is very interesting because these organs are heavily remodeled. So the spleen is enlarged, the lymph nodes are enlarged, the liver starts undergoing pathology, and it's also enlarged. So these I found interesting too, because when I was studying Plasmodium, we know that these organs are enlarged and damaged as well, and this has been related to if the parasite, so as a strategy of survival of the parasite. So if they go to these lymphoid organs and remodel them and in a way alter the capacity they have to respond to an infection, this is beneficial for the parasite in some ways. The third and fourth groups are more similar. So basically you see here that there are waves of intra and extravascular enrichment of parasites. And this is just to show the differences between the white adipose tissue and the brown adipose tissue. So here in pancreas and white adipose tissue, you see that almost all throughout infection, the extravascular reservoir is very, very high compared even to the intravascular. Whereas in the brown adipose tissue, it also comes in waves, but the intravascular population is higher. So those were the four groups we found. And so we went on to ask now what I was talking to you about before in terms of what's special about the vasculature of these different organs. So basically, the first thing we went to look at was whether so if you think about it rationally, is it that the organs that have more vessels are more enriched? So this would be a very straightforward explanation where it's something passive and it's more to do with the anatomy of the organ. So what we did was to quantify vascular density, and we did this like we show in this schematic. So you have a field of view of a specific height and width, and so there we can calculate the area of the vessel, for example here, and then say, okay, that constitutes say 20% of all of this area. And then we did this in a volumetric way, so we have the entire volume. And we represent it here in this heat map also as color coded. So basically the more yellow means that the vascular density is higher. So we have organs which have 80% vascular density. So basically most of the tissue is vessel. But as you can see here, we always group the adipose tissue and the pancreas at the top just because these were the organs of interest, because they're the highest reservoirs. So here you can see that they're basically not that high in vascular density compared to other organs like the brain, the lungs, and the liver, which are extremely vascularized, and yet they are not so highly invaded as the others. Then we have others that are low density as well, like the heart, the kidneys, and the brown adipose tissue. And then we have organs where vascular density decreases. And the reason why they decrease, or why vascular density decreases through time, is because there is a remodeling. So the vessel cannot keep up with the organ enlargement in the case of the spleen and lymph nodes. And so what happens is you have more tissue that is not vessel or not vascularized. Here in the white adipose tissue, we see that vascular density increases with time. And this is because these adipocytes start decreasing in size. So basically what you have is that the tissue is destroyed. And so the vessels are not retracting in the same rate. So you end up with a lot of vessels later in infection. But we still have to figure out whether this is active angiogenesis or simply because of lipolysis that we see this reduction. So basically finding 4 is that there is no correlation between organs that are more vascularized and organs that are most highly invaded by parasites. The next question we asked was whether this preferential location, still considering that it's a passive traversal, has to do more with the anatomy of the vessel, but this time in the sense of fenestration. So we know, for example, that in the liver, there's huge fenestrations, huge fenestra, where if things wanted to pass passively, it would perhaps be possible. We didn't know. And this is the total opposite to what we see in the brain, where the brain-blood barrier is very, very tight. So the way we found to measure this, and it's an established method, is to inject fitsidextran. So fitsidextran of a certain size, so you choose the molecular weight, and this might or might not pass through the vasculature, depending on how permeable it is. And so in a situation where the vasculature, so the vascular endothelium is very tightly joined, you would have no leakage of the dextran. It would remain in the vasculature and nothing would be outside. Whereas if there's some form of leakage or the junctions are not so tight, you would have dextran falling out into the extravascular side of the tissue. So it would look like this. So what we did was to measure the mean fluorescence intensity in the outside of the vessels. And so again, we represent this as this type of heat map, where the higher this mean fluorescence intensity is in the outside, the more yellow you would see it. So the first thing that became very clear is that all the organs become permeable with infection, maybe the heart not so much, but all the others, they increase compared to the initial situation that we see here. So the second thing that we see is that the pancreas and adipose tissue don't become, they're not special. It's not that they are much worse than everyone else. So for example, the brain becomes very leaky later in infection and still is not one of the most invaded organs. We then tried to induce vascular permeability to see if we could promote that there was more parasite traversal, and we did this by injecting histamine. So as you know, histamine induces so more permeability and vasodilation. And so again, if you see this little map, we saw that there was some higher increase in the liver, for example, but not anywhere else compared to the initial observations that we had made. So here you can see the comparisons. So basically, we saw some differences in the spleen, and in other organs, it was very similar within the initial days of infection. So the conclusion from here was that extravascular reservoirs do not correlate with high vascular permeability at this point.
Mariana,
yes.
Com licença, eu acho que você está tentando apontar na tela, mas a gente não vê.
Ah, ok.
Vai tentar ver se você consegue ativar algum modo diferente?
Yes. Otherwise, I will tell where exactly I'm looking into, because
Tudo bem.
Yes, I cannot find. Okay, I'll do that. Yeah, sorry. So in this part, what we went on to do was to look at the type of vessel that we're talking about. So we said, okay, if it's not just the anatomy and not vascular permeability, what about the type of vessel? So if it was an artery as opposed to a vein. And here on the left side of the panels where you see the images, it's simply what the methodology that we use. So basically you can see here how we subdivided the different groups. We called arteries anything that was above 20 micrometers. This is in general. It changes in some specifics among organs based on histology and anatomy that has been well defined, but we gave this blanket definition. So an artery or a vein is above 20 microns in diameter. An arterial or venule is between 10 and 20, and a capillary is defined as anything smaller than 10 microns of diameter. So based on this, what we did was we also needed a proxy. So basically there are organs which naturally receive more blood flow than others, more constantly. So what we did was to have something like a fiducial marker. So for this, we used exogenously labeled red blood cells with some form of dye, which you can see also on the left side of the panel. So the two figures that look either green, the red blood cells are green, or magenta. So we loaded those red blood cells exogenally, and then we introduced them into an infected mouse. And so now what we did was to compare relative to those markers if there was an enrichment of parasites comparatively. So on the right side of the panels, you can see what we found. So basically, again, 4 different groups of observations. And what we found was in group 1, which is where the white adipose tissue and the brain belong, we saw that there is an enrichment based on diameter, but not on type of vessel. So basically, both arteries, the arterial family and the vein family, they're equally enriched, but only in vessels that are higher than 10 or bigger than 10 microns in diameter. So not in the capillaries. The second group that you see, which is next to it, is the pancreas and lungs. And here we see enrichment everywhere. It's more time dependent than anything else. So you see that there's enrichment in the arterioles, in the venules, and in the capillaries, and in the same for the arteries and veins. So there it's more related to time of infection, and they are more enriched everywhere. In the third group, which is the first panel below, on the right hand side, here we see something interesting, which is the arteries are enriched preferentially and the veins are not. And this is the case for the heart, the kidneys, and the brown adipose tissue. And finally, in group 4, we have where there's no relative enrichment. So there are lots of parasites, but this is nothing more than what you would expect by random distribution, as what we see with the fiducial markers. So again, the conclusion of this finding is that this distribution is homogeneous. There seems to be some organs that are more enriched than others, and so this is worth further investigation. This is still at the vessel level, so they haven't crossed. We are trying to understand what happens before they cross and what explains the enrichment in the extravascular side of some of these organs. So having investigated these other things that were more related to passive, so something more related to the host rather than an actual active engagement between host and parasite. So now we will go to explore that. And so this is whether vascular endothelium and its receptors have anything to do with this preferential enrichment on these organs. And so we chose some receptors that we knew in advance were, so in advance as in other parasites. So I had done a lot of work in Plasmodium, and we had found some of these receptors to be, both me and others, that were important for sequestration. So we thought, okay, what happens in terms of infection and inflammation, and then in terms of traversal for T. brucei. So here at the top, again, you see in the first column, it would be the expression of the receptor in uninfected mice. And in the second column of each of the receptors, you would see what happens when the mouse is infected. So that's why they're all in pairs based on the receptor. So you can see that for the white adipose tissue, which is the topmost organ, something happens with E-selectin, something happens with ICAM2, although ICAM2 is very highly expressed in the adipose tissue, both in uninfected and infected mice. And so with VCAM to some extent, and with CD36 more obviously. And a similar pattern is seen for the pancreas. I was very excited with this project, but unfortunately the story that we published had to focus on something, and so we chose the white adipose tissue and the pancreas. But you can see that other organs, like the brain, for example, is super interesting, the lungs as well. There are several receptors like ICAM1 and ICAM2, which have a huge enrichment, CD36 as well. So there's plenty of things to continue to explore in that sense, but we focus the story, and so I'll continue talking to you about the white adipose tissue and the pancreas. So we chose those that we continued with all of these as you saw the differences. And so here, for example, in this slide, what I'm showing is what happened when we injected antibodies that block these receptors, all of the receptors that we found interesting. And so what we saw was that, so here, if you look at figure B, what happened when we injected E-selectin or P-selectin or P and E-selectin was a huge reduction in parasitemia during the first wave, so already early on. There was some reduction as well with ICAM2 and with CD36, but not as extreme as with the two selectins, which are known to work in junction with each other. This had an effect not just on parasitemia, but it might not be surprising to you that also mouse survival. So the mice that were that had E and P selectin blocked survived for forever, so well beyond 60 days. And then with other receptors, it depended. So with some there was quite long, 60 days, but the mice still died. With others, so that was the case of CD36, for example. With others like ICAM2, the reduction was more moderate in terms of survival, but still there was an increase. And so in terms of parasite load in these different locations, as you can see, so basically here in figure D, what you see in the heat map is the relative reduction. So basically a 0 is shown in white. So this is nothing happened compared to the untreated mouse. So this you can see the isotype control, which is the second column, is more or less similar to the untreated. But if you look at P and E-selectin together, they show a huge reduction, so a twofold reduction. This is in log scale, so this is 200-fold. 100-fold. And with CD36, you see some increase in spleen and liver, but a reduction in the white adipose tissue. So this was interesting to know that perhaps there are different players involved in terms of the pancreas and the white adipose tissue and what's controlling this traversal of the parasites. The other thing that we saw was that this here is related to parasite motility. So we saw that there is a huge change in terms of the preferred form of motion of these parasites when you block these receptors. So our hypothesis was that perhaps, or rather, when we were looking in intravital microscopy at the parasites, how they engage with the receptors in the pancreas and adipose tissue, we saw that the preferred type of motion was crawling. So the parasites go to the vascular endothelium and they start crawling on it. And so if these receptors are blocked, so P selectin, ICAM2 or CD36, what we see is a change in this type of motility. So here you can see that with P and E selecting together or alone, the parasites become immotile. So it's as if there's no traction, they cannot move. With ICAM2, it's similar to what we would have seen in the untreated control. And with CD36, they start tumbling. So again, it's as if they lose traction, but instead of not being able to crawl, they start just moving in the same location as if they were trying, but not being very able to. So that was interesting as well. And so this led to our model of more or less what's happening in terms of how the extravascular reservoirs are being established. So here you can see a summary of everything I said, considering that basically ICAM2, E-selectin, P-selectin, and CD36 play a role in the establishment of the adipose tissue reservoir. And this establishment is important for mouse survival and for general parasitemia. So then, as part of a second story, which we are still working on right now, we went on to explore tissue-specific properties. So basically asking the question, okay, so there's a part that is controlled by the vasculature, but once the parasites cross, and we saw, as I was saying during the presentation, that say there's a lot of crossing in the spleen or in the liver early in infection. So why do the parasites not form enormous reservoirs that can persist forever in these organs? And so this was more or less the question we wanted to address at this point. So what I started doing was seeing whether T. brucei distribute heterogeneously across the organ. So basically we were referring in the first story as the lungs or the brain or whatever. But the lungs and the brain and everything else have subregions. So the lungs have several lobes and different regions, so bronchi, bronchioli, and so on. The spinal cord has different regions and very much like this. So what we found was 3 specific patterns of distribution of parasites. So the first one is homogeneous distribution, and this is what we saw in these organs. So the brown adipose tissue, the liver, the spinal cord, and the lungs, where the enrichment is more related to time post-infection rather than to a specific region. So all liver lobes are equally parasitized, all lung lobes are equally parasitized, all the spinal cord and all the brown adipose tissue. And whatever enrichments, relative enrichment we see is more time dependent. So for example, the lungs are heavily parasitized late in infection, the liver is heavily parasitized early in infection. So that's one, homogeneous distribution. The second is a gradient type of distribution, and this is the case of the gonadal tissue, gonadal white adipose tissue and the pancreas, where what we saw was proximal to a large vessel. So imagine that the artery is entering the organ through here. Proximal to that artery, we would see more enrichment than we would see distal to that. So that's how the colonization begins, and then it starts spreading forwards. So at some point late in infection, one has the entire organ fully invaded. And the same is true for the pancreas. And then there is something I found very, very interesting, which is specific enrichment. And so in the brain, for example, there is a specific region which is very highly enriched in parasites, and other regions are not so much. In the kidney, this is the adrenal gland. So the adrenal gland is heavily, heavily, heavily colonized. The rest of the kidney, not so much. In the spleen, it's the red pulp, but not the white pulp or the marginal zone. In the bone marrow, it's the yellow marrow and not other places. And in the heart, it's the myocardium. So something is special about these locations within an organ that are more favorable for parasite colonization, whether the organ is a big reservoir or not. Other things that we found was that there's a huge heterogeneity in terms of parasite forms. So when we go and look into the parasites, they're not the same in blood so in blood they seem to be more similar to culture, what we can see in vitro. In the different organs, the parasites adapt a lot, and they change their shape, they change their speed, they change many things. So the first criteria we saw was stumpy formation is very different across the different organs. So here at the top, I'm showing based on percentage how it looks in blood. So basically, in blue, I'm showing the slender forms, and in green, the stumpy forms. So basically, you can see here the waves of parasitemia, which are of slenders, which are followed by waves of parasitemia in stumpies. But here you can see, and I mentioned before, that the spleen, liver, and red bone marrow are heavily colonized at some point early in infection, and you can see that there are no stumpies here. So this is very surprising, because there's all this idea that when the parasite population becomes very big, this is a trigger for switching into stumpy. So rather what we found here is that there's something about the organ that is also deciding whether switching to stumpy happens or not. The opposite situation can be seen in the brain, where at this point, so this organ is not extremely heavily colonized at any point, and yet it's quite enriched in stumpy forms all throughout infection. And then we have other cases which are more notorious, like the pancreas and adipose tissue, which almost in the entire course of infection are full of stumpy forms. Oh no, these are videos which I don't think you can see because I cannot see them. Anyway, what I wanted to show here was just that speed of the parasites changes a lot through time. So it's not only organ dependent, but also time dependent. So basically at day 7 and at day 9, the parasites are barely moving at all. At day 7, there's a decrease in speed compared to day 5. Sorry, these videos I don't think uploaded. But you can see this here. So basically here I summarize more or less what I just said. In the heat map, you can see the relative speed. So these numbers are microns per second. And so you can see that in the blood, the average speed is 20 microns per second, which is more or less what had been previously described. In the lungs, heart, and kidney, and brown adipose tissue, this speed remains more or less the same. So similar to what we see in blood all throughout infection. But then it becomes again interesting that other organs like the spleen, red bone marrow, liver, brain, pancreas, white adipose tissue, and yellow marrow, they have the parasites there are very, very fast at some points of infection. And again, the increases and decreases in speed are time dependent. So in days, for example, 8 to 12, there's barely any motility. They're super slow, and then they start increasing in speed again. So this is also a new wave of parasites. So we're not talking about the same parasites all throughout. So this is just to show that something in the tissues and with the parasite forms that we're seeing makes specific motility, so specific behavioral adaptations to the tissues. And so with this, I'd like to conclude that part. So I didn't show one big part, which is all the morphology of the parasite, so how they change and how we see much bigger parasites in some organs, and it more or less correlates with the speed. So basically, at some point, the parasites in the pancreas, white adipose tissue, and bone marrow are very, very long and very wide. In the brain, they're just very long, but not very wide. And in the spleen, red marrow, and liver, they're both long and wide. So these had never been described. The ones in the brain had been described, and it's consistent with what we see. So these long forms that just are like twice the length of a common slender form that we see in culture in vitro. So yes, as a summary of these parts, basically we found another important extravascular reservoir of T. brucei, which was the pancreas. We show that not all adipose tissues are equal reservoirs for T. brucei, that parasite distribution across the vascular network is not homogeneous, and that perhaps traversal is not passive. So it's not just the host that decides traversal, it's also this interaction between parasite and host with some receptors. Then we found some specific receptors that are important for reservoir establishment, that is ICAM2, P, and E-selectins. And then CD36 seems to be important for adipose tissue establishment. And although we didn't investigate it, the lungs also show an important increase in CD36 expression. And in the second part, which is something that we are trying to publish at the moment, is more related to the behavior and the morphology and the adaptations that the parasites display in the different organs. So basically, what we are saying overall in this side is that the parasite population adapts to the different organs. It's not the same population that then we see in the blood. And so with this, I want to just briefly tell you in like 10 minutes what the work I did at Institut Pasteur, maybe even less than 10 minutes. So basically, I went there for the purpose of learning electron microscopy. So I had dedicated most of my career to work on fluorescence microscopy, optical microscopy, and I felt that something was missing and that I wanted to really get hold of electron microscopy too. And then we started with different projects. One project that derived from the work I did in Luisa's lab was this one. So basically, we wanted to be able to see parasite motility and its behaviors at a single cell level. But of course, if you've ever tried to image T. brucei in vitro, it's almost impossible unless you squeeze it in between slides because it likes to swim a lot, and it swims very randomly and very fast. And so it's very difficult to image. So what we thought of was to make some form of trap where we could trap the parasite in a specific location and keep it there in a confined space where we could image it without completely preventing it from swimming and without putting a cover slip on top of it or using a chemical way of blocking its movement. The usual is polyalysine. So we generated a microfluidic device, a little chip, where we tested different types of traps, and we ended up finding this particular one, which we are in the process of publishing as well. It's this V shape that seems to be, they like going into the corner, the parasites like going into the corner, and then they start doing different types of motion within it, but they're free swimming, so they can choose to leave whenever they want, and so you are able to see a lot of the movements of the flagellum and to follow that same parasite for a very long time. So we managed to watch them for 8 hours, and 8 hours is enough to watch an entire the cell cycle progression, so in one round. The second project I did in Philippe's lab, and now it relates to electron microscopy, was this one. So basically, we generated some FIPSEM, and so I don't know if you're familiar with Philippe Bastan's work. He likes intraflagellar transport train. He focuses on that, and so intraflagellar transport trains are trains within the flagellum that transport different proteins, lipids, and so on within the flagellum. And so my interest has been more throughout my career as well on these other organelles, so ER, Golgi, and mitochondria. I've studied them in a different context, so always from the host side and how they're hijacked by parasites. But here what I found interesting is, okay, so if we abrogate intraflagellar transport, what happens to these membranous organelles? So you always see, when I started looking at this FIPSEM series, I saw, wow, the Golgi, for example, is always proximal to the flagellar pocket. So you always see it like this. If we look, somewhere else lower in the FIPSEM series, you would see that, the Golgi is very close all the time. And it remains close to the flagellum all throughout the series. And then the ER is close to the Golgi, and the mitochondria is close to the Golgi. So they're all, together with many contact sites happening. But what happened when we make IFT mutants? So basically, there is no transport of anything going through the flagellum. What I found, which was quite exciting, is that that all these membranous organelles become big. So it's as if they start accumulating. So nothing is getting transported into the flagellum, and so the organelles grow in size. And they distribute, instead of being just proximal to the flagellar pocket, you can find the pieces of Golgi all over the body of the parasite, which is what one can see here, or more or less. So here what I wanted really to do was to learn how to use not just sample preparation and do the FIPSEM, but also all the image processing and image analysis. So basically doing the segmentation, the reconstructions, and then being able to reach conclusions. So we're working on that still now. But in general, the conclusion was this, that interfering with intraflagellar transport causes a swell-up of membranous material that accumulates in these organelles. And the last and probably most important project that I did during this time was, so Philippe is interested in intraflagellar transport trains, and the way they look is little dots that are around. So basically, yeah, I cannot use the pointer here, but yeah, they are little dots that you can see next to the beyond the paraflagellar rod. And the structure I was interested in was the paraflagellar rod itself, which is the biggest structure that you see in the second image of the flagellum, so a TM image there. So this structure, basically what I did was to generate mutants that lack the paraflagellar rod. And something interesting that I saw that I came back to relate to the work I did with Luisa Figueiredo was to, when I was looking at these parasites, at the mutants in vitro, I saw that they cannot do the whole range of movements that the wild type parasites can do. So the question that it brought for me was, what if, so one of the things that I anyway wanted to do was to systematically generate knockdowns of parasites with different flagellar components to see how that affects tropism. And so my first finding was that this paraflagellar rod is important for the corkscrew type of motion that the parasites can do. And the hypothesis is that this will be important for traversal of tissues. So now I just have to continue. And Philippe was like, yes, you can take this project and investigate it in the context of what you like to do. And so with this, I would like to thank the different labs, so Luisa Figueiredo and her entire lab that were always very, very supportive. The bioimaging facility at the Institute of Molecular Medicine is excellent, so I had a great time there. There are a huge amount of microscopes. Claudio Franco was a collaborator that is a specialist in vasculature, and so him and some members of his group were very helpful. Nuno Santos is another PI at the Institute, so he works on biophysics, and with him we did some work that I didn't include in this presentation, which was mostly related to the viscoelastic properties of the tissues and how that might explain how the parasites move once they have crossed out of tissues. And then the rodent facility, so Yolanda and Daniel, who were the in-house vet and the rodent facility manager. So as you can imagine, we used several mice. We used several colonies of different types of mice, knockouts, reporters, and so on. So they keep them always. And then at Institut Pasteur, Philippe Bastan and his lab that taught me how to do electron microscopy. And the fellowships that funded my work, EMBO and Human Frontiers, and you for your attention, and Angela for the invitation, of course. And with this, I'll take any questions.
Obrigada, Mariana. Deixa eu ligar nossa luz aqui. Muito obrigada, fascinante sua palestra, a forma como você transitou, né, como o ambiente modela o parasita e depois em detalhes moleculares, muito legal mesmo.
Muito obrigada.
Mariana, parabéns, muito obrigada. Eu tenho eu teria muitas perguntas, mas vou deixar aí que tem outras pessoas, né? Mas, assim, uma coisa que eu fiquei pensando foi, quando você tem os parasitos no tecido adiposo branco, em torno do pâncreas, vocês chegaram a ver, em algum momento, se esses animais desenvolviam diabetes?
Isso é muito interessante, claro. Então, a Luísa está interessada na doença metabólica. Acho que o grupo dela está interessado mais no que é a lipólise, que o animal fica magro, e entender mais ou menos o que ocasiona que o adipocito fique pequeno, que tenha uma libertação de lipídios da lipid droplet. Mas eu realmente acho que ela não estava muito interessada no pâncreas, e para mim é muito interessante. Então, realmente, sim, eu acho que isso é algo muito importante. É uma resposta que queremos procurar depois.
Então, você está associando com perda de peso ou ganho de peso, alguma coisa assim também?
É uma perda de peso muito grande, sim.
Ah, sim. Eu pensei nisso, Porque pâncreas, né? Teria tudo a ver, né?
Sim, exato.
Então, enquanto nossa plateia está aqui meio tímida, vou te fazer uma pergunta. Eu sou fora da área, não sei os detalhes, mas é possível tentar um tratamento e ver como é que isso afeta esses reservatórios? Tem algum local que funciona? A gente vê que a biologia do Trypanosoma está toda alterada, né? Isso pode induzir resistência a alguns órgãos, enfim.
Sim. Uma coisa que eu estou interessada é precisamente na linha da pergunta da Ângela, que é que acontece, ou seja, mesmo que seja possível eliminar totalmente os parasitas com algum medicamento, os órgãos, desde o início, assim, imagina, o fígado, desde um dia 3, já está bastante danificado. Então, uma pergunta que eu queria responder, já como cientista independente, seria qual é o efeito na saúde a longo prazo pela essa remodelação e esses danos nos diferentes órgãos.
Uhum.
Então, sim, certeza.
Muito legal.
O que eu achei também surpreendente foi como que os Stump e os Slender têm tropismo por órgãos diferentes, né? Isso eu nunca tinha visto isso.
Sim, é um pouco mais. Então, essa é uma outra pergunta que agora, neste paper que estamos a fazer, estamos tentando resolver, que é alguns slenders que têm já aquele marcador PAD1-GFP, que é o reporter ou seja, está maximamente expressado no STUMP, é realmente um marcador de commitment, ou de diferenciação, que esse parasita slender já vai para ser STUMP. E essa é a pergunta também, que há tanto migração de STUMP ou de STUMP committed parasitas para um órgão qualquer, ou que um slender, quando encontra um ambiente, por exemplo, no adipose tissue, é exposto a alguma signaling ou algum environment que já deixa ele mais propenso a se converter em stumpiform. Isso é outra coisa que estamos a investigar bastante.
Você falou de receptores, né? Nessa. Mas vocês estão vendo também os receptores no parasita?
Sim, então isso é uma coisa que a Luísa está investigando, acho que com a Érida.
Já ouvi falar, sim.
Acho que ela é brasileira, não?
Eu já ouvi falar, mas eu não estou ligando,
Sim, sim, elas estão investigando o SOR-film, eles chamam. SOR-film do parasita. Qual é qualquer ligando que se engage com os receptores, aqueles que encontramos que eram importantes. Mas até agora não sabemos, não.
Muito legal. Mariana, mais uma vez eu agradeço você ter aceitado prontamente o nosso convite e a ciência muito linda aí por essa hora. Obrigada por ter dividido isso com a gente.
Obrigada também por a gente poder ver você, porque eu conhecia você muito de e-mail, né? Mas eu nunca tinha visto você.
Sim, igualmente, sim. Muito obrigada pelo convite.
Pessoalmente, em breve.
Sim, exato. Espero também conhecer em breve. Muito obrigada.
Obrigada a você.
Tchau, pessoal. Até o próximo seminário.
Tchau.